Today we're going to teach you about bacteria and their plasmids and how plasmids are used to make recombinant insulin. But what's a bacterium? We'll explain it to you. A bacterium is a prokaryotic microorganism and are just a few micrometers in length.
Bacteria have a number of shapes, like spheres, rods, and spirals. Because they're so small, they can be found anywhere. Oh look, it's Mr. Bacterium. He's going to assist us today. As you noticed, he's covered in little hairs we call pili. They make sure the bacteria can exchange DNA by connecting each other's cytoplasm. He also has a flagellum, so he can move around. The inner structures are protected by a mucus layer we call the capsule. If we cut Mr. Bacterium in half, we can see all different kinds of structures. On the outside, we have the cell wall and the cell membrane. As you can see, the bacterium is filled with cytoplasm, which contains nutrition to feed the cell. Within the cytoplasm, the ribosome can be found. They play an important role in the buildup of proteins. Then, we have the chromosome and the plasmids that contain the DNA of the bacterium.
Plasmids, you say? Let's talk about those.
A plasmid is a small DNA molecule within a bacterium that is physically separated from the chromosomal DNA and can replicate on its own. Plasmids are usually very small and contain only additional information. But how are they used to make recombinant insulin?
Recombinant DNA is a technology which made it possible to insert a human gene into the genetic material of a common bacteria. The microorganism can now produce the protein inserted with the human gene.
First, the scientists remove a plasmid from the bacterium. Then, with the help of a restriction enzyme, they make a cut into the loop of a DNA. After that, they insert a human insulin gene derived from a genomic database. The human insulin gene connects with the rest of the bacterium's DNA to close the loop again. The engineered plasmid is now inserted into a new bacterium. The new bacterium is then put into a large stainless steel fermentation vessel so it can divide and produce insulin. But what does insulin do?
Insulin is a hormone used to regulate the metabolism by storing the abundance of glucose extracted from the blood into fat tissue and muscle. The fat tissue can later be used when you need a lot of energy, for example when you exercise. But why do we use this technique? When you do not produce enough insulin, you suffer from a disease called diabetes. The disease can't be cured, but it can be treated by injecting the insulin shots derived from the recombinant DNA technique. That was today's lesson. I hope to see you next time.